Hi, my name is Kylie Nee. Welcome to my presentation on the reproductive physiology. Specifically, I'll be talking today about the regulation of sperm maturation. So just a little overview of what we'll be talking about today. So first we'll be talking about the vocab that's important to know, and then we'll be talking about sperm production, the hormones that are required for spermatogenesis, and the secretion of semen. So just some important vocabulary to know before going into this. So first is the testes or the testicles, which are the male reproductive organ. There's also the seminiferous tubules, which are the site of sperm production, and they're clustered into 250 to 300 compartments that are enclosed by the tough fibrous capsule of the testes. There's also spermatogonia, which are the germ cells that undergo meiotic division to become sperm, and they're within the seminiferous tubules. There's also the Sertoli cells, which are also within the seminiferous tubules, and they regulate sperm development and provide sustenance or nourishment for the developing spermatogonia. And then there's also the interstitial cells, which secrete testosterone. So the seminiferous tubules seem to be a little important. So we're going to go into a little bit more about the seminiferous tubules. So the seminiferous tubules contain two types of cells, the spermatogonia and the Sertoli cells. And the spermatocytes stack into columns from the outer edge of the tubule to the lumen, and a single Sertoli cell extends from the outer edge of the tubule to the lumen. Surrounding the outside of the lumen is the basal lamina that acts as a barrier, and this prevents certain large molecules in the interstitial fluid from entering the tubule, but it allows testosterone to enter very easily. The Sertoli cells rest on that basal lamina, and this creates a basal compartment. And there's also something called the blood-testes barrier, which is a tight junction that forms from adjacent Sertoli cells that are linked together, which create an additional barrier between the lumen of the tubule and the interstitial fluid outside the basal lamina. So now let's talk a little, so these are just some pictures showing the seminiferous tubule. So on the top, you can see a cross-section of a seminiferous tubule. So you can see those interstitial cells that secrete testosterone in the corners. You can see a spermatogonia right here. They're all over the place. And then you can see those purple Sertoli cells over here. And then they just took a little zoomed in picture of this part of the seminiferous tubule, and this is right here. And this just shows sperm development. So we can see those spermatogonia again. I'm sorry, I don't know why this keeps popping up. And then you can also see that tight junction between the Sertoli cells, and we can see that sperm development goes up into the lumen. So from the germ cells to the mature sperm, it goes upwards. So now we'll talk about sperm production. So the spermatogonia undergo mitosis in the basal compartment. So some spermatogonia will actually remain in the basal compartment, while others will begin meiosis and become primary spermatocytes. And then they'll move inward towards the lumen of the tubule, and they're surrounded by Sertoli cells continuously. The blood-testes barrier will break and then reform around those migrating cells. And then by the time that they reach the luminal end, they'll actually have divided 2 times and will have become spermatids. And they'll remain in the apical membrane until a complete sperm transformation happens. And so the chromatin of the nucleus condenses into a dense structure that fills most of the head, while a Golgi-derived vesicle called an acrosome flattens out to form a cap over the tip of the nucleus. And the acrosome contains enzymes essential for fertilization. So we can see over on the right, this is a picture of a sperm, and we can see that acrosome cap right here. And so mitochondria produce energy for sperm movement concentrate in the mid piece of the sperm body, along with microtubules that extend into the tail. So once again in this picture we can see that mid piece in the tail right here. Otherwise it's also known as the flagellum. Sperm are released into the lumen of the seminiferous tubule along with secreted fluid, and from there they are free to move out of the testes. The entire development process from a spermatogonia until sperm release takes about 64 days. And the staggering development of these stages allows sperm production to remain nearly constant at a rate of 200 million sperm per day, which actually isn't that much because it's about the number of sperm released in a single ejaculation. Sperm just released from the Sertoli cells are not yet considered mature and are incapable of swimming, and they must complete their maturation during the 12 or so days of their transit time through the epididymis, and their maturation process is aided by protein secretions from epididymal cells. So now we're going to talk about the hormones that are required for spermatogenesis. So hypothalamic GnRH promotes release of LH and FSH from the anterior pituitary, and this stimulates the testes. So FSH targets Sertoli cells, and this is needed for spermatogonia mitosis and spermatogenesis, and stimulated production of androgen binding protein and inhibin. So then the LH targets interstitial cells, which then triggers testosterone to be released, and this feeds back to inhibit LH and GnRH release, and testosterone will be mediated by those Sertoli cells. So over on the right, we can see a figure that shows the hormonal control of spermatogenesis. So starting at the hypothalamus, it then goes down into the GnRH, and then that triggers the anterior pituitary to release FSH and LH. And you can see that the FSH will trigger those Sertoli cells, and the LH will trigger those interstitial cells, and then testosterone will be released, and that goes to the body for secondary effects. And then the Sertoli cells will trigger those cell products like inhibin or the developing sperm, as well as the androgen binding protein. So finally, once those sperm are mature, they'll be secreted. So now we're going to talk about the secretion of semen. So there's 3 accessory glands which have the purpose to secrete various fluid mixtures. So there's the bulbourethral glands, which contribute mucus for lubrication and buffers to neutralize the acidic environment of the vagina. And then there's the seminal vesicles, which contribute prostaglandins that influence sperm motility and transport in both male and female reproductive tracts. And then there's the prostate gland, which contributes nutrients for sperm metabolism. And then the sperm is actually joined by these secretions during ejaculation, and this results in a sperm fluid mixture, also known as semen. And so 99% of the volume of semen is actually just those fluids added by the accessory glands. So 1% of semen is sperm. So there's not a lot of sperm in semen. And then the accessory gland secretions not only help with sperm motility, but also help protect male reproductive tracts from pathogens that may ascend the urethra from the external environment. And so once those mature sperm cells get to where they need to go, the rest is kind of history. So thank you for listening to my presentation. Bye.